part of ice cream is really that experience of the ice cream melting in your mouth. We can fundamentally change that with these antifreeze proteins, which we basically are getting because a bunch of nerdy biologists are like, hey, why did that fish not freeze? It's really great to be back. This is such a lovely community to chat with. So this is not my area of expertise, although I saw that you had my paper up there, one of my papers up there. But it's a super cool area, and we should end up with lots of stuff to talk about. Sure. My name is Thomas Merritt. I am a professor at Laurentian University. I have been here since 2006. I am a full professor, and I use Drosophila mostly to study the connection between genetic complexity or genetic diversity and biological complexity is the way I usually spin it. But we ask lots of questions about how individual variation impacts the way that you respond to the world around you. I do a lot of science outreach, and a lot of science outreach is finding a topic that people find engaging and then use that to talk about things that are really interesting. There are a couple of things I want to talk about today. I mean, we'll go through a bunch of different things, but
one question that we just had to start off the stream was, what is a cold shock and heat shock protein?
Okay, so heat shock proteins are named, so I'm not a great history of science guy, but heat shock proteins are named because traditionally we would take a model like a mouse or a fly and we would heat it. Like literally, we would put it in a warm environment and we would look to see, I mean, this is the classic poke it and see what happens thing, right? So we would heat the system and then we'd see these series of proteins that would go. Heat shock proteins tend to be small. And they're called heat shock proteins just because those are the techniques that we use to study them. They're proteins that respond to changes in the environment, often temperature, but they can be other things as well. And this is off the top of my head, but I think that there's a parallel between things that have been classified as heat shock proteins and proteins that are involved with stabilizing a system or stabilizing other proteins. And so maybe things like chaperones, which are involved with protein folding, those have been sort of tied in. So it's a big umbrella class, literally named because of the high-tech science of, I don't know, what do you think will happen when we heat it? Which is the way that we all do science.
And was cold shock named the same way, or is that just a historical?
Yeah, I think it's like the other side of it. And even in that, so the paper that I sent you that I'm on, that was really driven by Heath McMillan, was a grad student who's now a faculty member at Carleton. And he's going to give a talk for me in a class I'm teaching on bioinformatics in a couple of weeks. He's super. He's an amazing guy, fantastic scientist. So he was doing this as his graduate work, and Brent Sinclair was his PhD advisor. So the two of them have a very long-term history of looking at temperature acclimation. And I think the cold shock was like, we can't keep calling things heat shock proteins when we know they're not actually involved with heat. The really cool thing with Heath's paper, and the reason I wanted to take a look at it is as funny as it is to talk about things like we're gonna poke something and see what happens. We learned an amazing amount by taking that really sort of brute force approach. But one of the things that we learned was that if you are more biologically appropriate in the way that you poke the bear, you'll learn more about how a bear really behaves. And so what Heath and Brent were doing with that paper is they were cold acclimating flies. And so one of the things I was actually talking about class this morning was, what's the system that you're working on? And I mean, you've got a history in Drosophila. If somebody had asked me, what's the system that I'm working on, I would have said Drosophila melanogaster. Like, oh, okay, that's a piece of it, but what else is involved? And defining the system is a much more thoughtful process than just saying I work on fruit flies. So what Brent and Heath had realized was that if you slowly and long-term and less extreme stress an organism, you get a different answer than if you do a really acute stress. And so we take a similar approach in my lab when we're working with toxicity. And what I often say to my students is, what I want to study is a stress effect, not a death effect. So, the way that things die and the way that you respond to dying is really interesting, but it's not the biology that I'm interested in. So when we do things like nickel stress, will use a reasonably low concentration and a longer period of time. And so what Heath and Brent were doing with this paper is they're using cold for a period of time to really acclimate the organism to being cold, as opposed to stressing the organism with being cold. That stress tells you something, but it tells you something different from acclimation. One of the things that we always do in scientific papers is we present our results and then we reflect back on the results that led up to that. And one of the interesting things in Heath's paper was how different his results were than some of the expectations. And I think what came out of it was a realization that those expectations were based on these really short, acute stresses and not the longer term stress. And the other thing that Heath was doing was working in adults. And the one thing, and we should come back to this, and the other thing that we did in this paper is we worked with adult males. And that's a really narrow cross section of the world. And if we were to do this paper now, so this was published in 2016, we probably did the work in like 2014. So if we did the paper now, we would take a different approach. We wouldn't take that narrow focus. We'd probably say Drosophila melanogaster, but we do males and females, because what we've realized in the last 10 years is if you want to understand biology, you can't simply say, well, I'm going to look at males and assume that females are similar, because the biology is often very profoundly different between.
And that's actually some of what we've talked about in a couple of different past chats was, it's even become, like you were saying, a mandate in certain grants as well.
Yeah, 100%. It's less rigorously controlled in Canada. So I'm reviewing grants right now for INSERC, which is our federal organization that funds basic science. And I reviewed grants 3 or 4 years ago, and I've been away for 3 years. I've come back. One of the things that we're looking at differently, even in that 3-year window, is now if your grant should include males and females, it needs to include males and females. If it doesn't, say if you're working on something that's male specific or female specific, or if you're working in an organism that doesn't have males and females, it doesn't have to. But part of the grant review process is essentially if things like some aspect of diversity needs to be included, isn't included. And if it's not included, you can actually be counted off in your grant reviews for that. So if I were reviewing that paper that we have in front of us now, I would say this is really interesting, but either you need to argue to me why you only use males, or you need to repeat the study in males and females. Because if we want to say cold acclimation wholly reorganizes the Drosophila melanogaster transcriptome and metabolome, that title would literally have to be in males.
Yep.
So the other part is it would have to be in adults. We're not, at least at this point, as caught up on whether you're studying all life cycles or life stages in the life cycle of the organism. But we are very clear that if you are studying an organism that has males and females, you can't just arbitrarily say, we're only going to study females, or we're only going to study males.
With this paper and this kind of finding, there's easily to make an analogy to humans, right? Like how you can adapt to certain heat or cold. How adaptable do you think that this paper is to some, like the findings to humans, and what does it mean?
Yeah. So how can you extrapolate out from this? That's a really great question. And this is part of that model species conversation. One of the things that I found interesting in this study was that depending on how you sort of parsed out the different things we were looking at, anywhere from 20 to 30% and maybe 50% of the things that we were looking at, so whether it was genes or metabolites, were responding to a change in the environment. And it ends up that percentage, somewhere around 30-ish%, is reasonably constant. So we did a set of experiments that we're still struggling to publish, where we took flies deep underground into a mine environment. And as you go further underground, the pressure goes up. So you've got more atmosphere on top of you. And so this was a temperature-controlled mine environment. It's actually Snow Lab, which is a deep underground neutrino observatory. And for our flies, what was changing wasn't the temperature, but it was the atmospheric pressure goes up about 22%. And when we did that, about a third of the metabolites changed in response to that difference. So there are a couple of things that probably really do extrapolate really easily. Like changes in environment seem to, across different kinds of changes, impact somewhere around a third-ish of your metabolism, which is an interesting number. Like it's not the majority, but it's not a little fraction. So when I started the project, I was thinking, I don't know, maybe 10% of the metabolome or 10% of the genes. And it looks like environmental changes hit a larger percentage than that. And that, I think, extrapolates. Some things do and some things don't extrapolate. So flies are not, they don't thermoregulate. So they basically take on the environmental temperature. So their response to temperature is going to be different than our response to temperature. So tomorrow morning, it's supposed to be about minus 30 here Celsius, which is going to be, minus 18, 20, something like that. I'm going to go for a run. And this is like one of the things I do. When it gets cold, I want to make it a point to definitely go out and get out for a run. So like the coldest I've run at is minus 36. And I can tell you that there is a noticeable difference between minus 36 and minus 30. Like I went out for a run at minus 30 one day, a couple of days later it was colder, like I'm definitely going for a run. And I'm running, I'm like, oh man, you can really feel this difference. And so the way that our body responds to that cold, because we thermoregulate, there are a whole set of things that are going to go on that are not going to happen in a fly. There are some other things that are going to happen in a fly. And so there seem to be some things that are consistent when we look across models. So how much do the individual details change in the same way? That's a really interesting question. And basically what you have to do is look at a number of different systems and say, okay, in this species, in this species, in this species, do we find consistencies? And so there are some consistencies in patterns of amino acid metabolism that seem to be consistent. And there are a lot of things that are not going to be consistent. I think the broader picture, like what percentage of genes change in expression, what percentage of metabolites change in concentration, I think we've got a better chance that those answers are reasonably consistent across different models, different species. This is an interesting study. It basically looks at if you've got an organism that is normally in this environment, what happens when you move it to another environment? And the thing that Heath and Brent did here that I think is really clever and really informative is it's not an acute short bang, it's an acclimation. And so we're looking at how things acclimate to the cold, but we're looking at things on an individual basis in a population in real time. Okay? The other way to think about cold biology is how different are organisms that have evolved to live in the cold compared to organisms that haven't evolved to live in the cold. And so a lot of those studies, a lot of the early studies were basically driven by the fact that people were finding fish in areas where the seawater was below the freezing temperature of fish. And so you've got this physical inconsistency. So seawater doesn't tend to freeze because it's really high salt. And one of the interesting things about being a fish is whether you live in salt water or live in fresh water. So if you are a fish living in salt water, your osmolarity is lower than the world around you. If you're a fish living in fresh water, it's reversed. And there are really interesting patterns of what happens to a fish that moves from saltwater to freshwater, from freshwater back to saltwater. And this is something that we don't have to face. Like we're just these old bags of water with this, seal around us. And unless you sit in the tub too long and get sort of pruny, you're not really thinking about how your osmolarity compares to the world around you. But this is absolutely physiologically something a fish has to do because it's actually the environment is coming through the fish. And so the fish is either going to diffuse out into the environment or the environment's going to fuse into the fish, which is a really horrible way to think about osmosis. But basically, the osmotic pressure is going to be reversed whether you're a freshwater fish or a saltwater fish.
Yep.
Okay? So freshwater freezes super easy at the magic number of 32 if you live in America. If you live in the rest of the world, it freezes at 0, right? So people realized they were out in these environments that were saltwater, and the water was below 0. Like, okay, well, that's because it's saltwater, but the fish are not as salty as saltwater. So the fish should be solid. Like they should be cubes, and they're not cubes. And people are like, that's cool. How can the fish stay mobile and be below what should be its freezing point? And so over the course of the last, this is like 75 years. I think the original studies, there are probably studies that are older than that, but the studies started coming out in the 60s through the 80s, describing these antifreeze proteins. So one of the cool things that's come out of antifreeze protein biology is that there are lots of different antifreeze proteins in different fish that evolutionarily have no relationship whatsoever. And functionally, they keep the fish from solidifying, but evolutionarily, there's not a common ancestor. And that's one of the punchlines with those blind cavefish, was that even when we looked at these Mexican canefishes where you've got multiple cave systems, and you've got a blind population here and a blind population here, at least at first pass, it looks like there's a different switch that's missing in this population than that population.
I guess going back to the comparison that you were making of like being able to adapt because of the environments you live in versus having a particular adaptation because of a constant environment that you're living in. Right. How does something like that end up like evolutionary pressure wise going in either direction? Like what's the benefit to being able to freeze versus just being able to be like the fly, which has that range of adaptation?
Well, and so it's interesting because flies can't freeze. So there are a lot of organisms that can be cold tolerant, and you can. So the point of Heath and Brent's paper was that actually the fundamental bottom line with their paper was that you can change cold tolerance. And so the reason that we were looking at changes in gene expression and metabolite concentration was this observation that if you take a set of flies that are adapted to a temperate environment, they go into a cold coma at this temperature. If you acclimate them to the cold, they now only go into that cold coma at an even lower temperature. I think it's about 3 degrees Celsius lower. So you're actually you're acclimating to that environment. If you go below that, they just die. So there's cold acclimation, but there's no freeze tolerance. The cricket that you're talking about and a lot of other organisms, strikingly the goldfish, are freeze tolerant. And so you can freeze them solid. They're immobile, you thaw them out and they hop along or swim along. So it ends up there are different ways that tolerance has evolved. And so when we talk about antifreeze proteins, there's sort of two major classes. There are proteins that keep things from freezing, and those are the proteins that were found in things like the Arctic or Antarctic fish, and they're things that allow things to freeze. So if you freeze, the biggest issue with freezing isn't the fact that you're frozen, it's the fact that the ice crystals just absolutely shred the tissue. So you get this formation of crystal structures. The crystals literally sort of expand, and then they just cut through the cells. So what happens in freeze-tolerant organisms, at least in some, is that there are these proteins that allow the ice to nucleate around the protein, and they prevent these large crystals from forming. So in general, antifreeze proteins or ice-binding proteins bind to ice crystals and keep the large crystals from forming. In one class of antifreeze proteins, the animal doesn't freeze. And in the other class, you have freeze tolerant. And what happens is the organism freezes, but it does so without shredding the tissue because those large ice crystals don't form. So those are two different evolutionary solutions to this problem of what happens when you go below the organism freezing point. Those two sort of master switches are evolutionarily distinct, and there's no single switch either way. So there are a series of evolutionarily unrelated proteins that allow unrelated organisms to freeze and then thaw and go back to the living. And there are a series of unrelated proteins in a diverse group of organisms that allow those organisms to not freeze at a temperature below what would have been their freezing. Right? And so that the first case, the frozen case, is pretty straightforward. The organism freezes, it's immobile, it goes into some sort of coma, essentially, and then it comes out of that. The other case is the organism happily just lives on, but it does so below the temperature that it should be frozen. If you go below a certain temperature, it's going to freeze. So it's not magic. It doesn't keep you from freezing. It keeps you from freezing at what normally would be your freezing point.
So how does this work in cells? Do they do something to the water that prevents it from crystallizing, lowering the temperature of the water, or do they do something to the cells that prevents them from being damaged by freezing?
Yeah, so both of those. So in the case where things freeze solid, it allows the cell to just freeze, but it freezes in such a way that you don't form large shredding ice crystals. In the other case, these proteins nucleate ice and allow the formation of small ice crystals, but not big ice crystals. But it never solidifies to the point where that organism is in solid. And again, this is, Not exactly, not even exactly. This is not my area of expertise, but this is my take on it. And the idea of the antifreeze proteins has been something I've been interested in for the last 20 years because it's such an interesting evolutionary story, having evolved independently. A lot of the early work that I knew about was in fish. And initially, we talked about there being two categories of antifreeze proteins in fish that were structurally distinct. Within a category, they might be evolutionarily related, although it could be a convergence on structure. The paper I was looking at this afternoon, they're up to like 5 categories. They're like, yeah, we thought it was 2, but then we looked at these fish and we've got this category, then we added this fish, we added this category. And then the next sentence of the paper I think is awesome because it basically says, and then when you leave fish, things are just a wild west. There's so many different antifreeze proteins outside of fish that the idea of can we categorize it, you're chasing your tail. So there are at least dozens of different proteins that have evolved to allow an organism to not freeze at a temperature below what would have been its freezing point. And there are probably an equal number of systems that will allow an organism to freeze and then come back from that. So the application thing, though, absolutely, there are lots of really interesting applications. I think it's unlikely that we're going to be trying to evolve some kind of Aquaman who can swim under the frozen Arctic because we've infused them with antifreeze proteins. But there certainly are potential applications, and one of the issues is you never know where the applications are going to come from. And so a reason that we do undirected fundamental research is that we find interesting things that later can have applications. So, I think one of the universals that everybody can appreciate is like a good scoop of ice cream. And I think if you asked, what defines a happy life, then a ridiculous proportion of the world would say, I think what defines a happy life is a good scoop of ice cream. Well, it ends up that cryobiology is part of creating a better scoop of ice cream. So, and I'm not even making this up. I would have if I didn't have a better story, but this is such a good story, I don't even have to make something up.
And just as an aside, we actually had a question from chat of, can you eat these proteins or put them in an ice cream? Before you said that.
Yeah, okay, so 100%. And I knew this was out there, so when I started looking for some papers to play with, the paper I didn't send you was eating silkworm larval protein. And I was like, oh, that looks really interesting. And then I read the whole paper. And at the end of it, basically, we had a bunch of silkworms. We were trying to figure out what to do with them. So we just looked to see if it was toxic, if you could eat that. It was no tie whatsoever to cryobiology. It was just like, we got a bunch of silkworms. What could you possibly do with this? So if you think back to the way I was describing the way these antifreeze proteins work, they work by changing the way that ice crystals form. So they form these little nuclei, and they form smaller ice crystals, and they prevent large ice crystals forming. And one of the ways you can define a really bad scoop of ice cream is if it's really crispy, right? So I think we've probably, maybe not all of us, but many of us have gone for that tub of ice cream that's been in the freezer just a little too long. You're like, it'll be fine. You scoop it out, like, this is horrible. And what's horrible is that because your freezer does these defrost things, maybe because you're standing there with the freezer open trying to figure out whether you want another ice cream, that ice cream has gone through these freeze-thaw cycles. And that's promoted formation of large ice crystals. And so I think this is actually in practice. There are antifreeze proteins that are, at least initially, they were derived from other organisms. There's actually a note in one of these papers. And when you add these proteins, I think it's an insect protein, to ice cream, you get better mouthfeel. And basically what that means is the ice crystals are happier, they're smaller, and they make a more pleasant ice cream if you add this antifreeze protein to the ice cream. So there are absolutely applications. I think there are interesting potential biomedical applications. I don't know of any that are in practice. Doesn't mean that they're not. I just don't know them. But what I can tell you is absolutely 100%. They're already being used in food processing. And the other thing that they've been used for is one of the issues with frozen foods is the texture of those frozen foods. So with ice cream, it's like the defining characteristic of ice cream is that mouthfeel. But if you're going to freeze carrots, you're going to freeze meat, you're going to freeze potatoes, anything that you freeze, oftentimes the texture changes with freezing. And so they've been looking at how you can add in antifreeze proteins to reduce formation of these large crystals. So the proteins evolved literally to keep the large crystals from shredding tissue when a cricket freezes, but we can co-opt those to make a better freezing bread dough.
So is that a GMO? Kind of to tie us back to our very original discussion, right? Is that something that would be considered a GMO? Like this carrot, if I freeze it and unfreeze it, it's going to texture-wise be the same as before.
Yeah, so depends on how you're playing the game. Like, could you genetically modify a carrot to express an antifreeze protein? 100%. Would it be a genetically modified organism? 100%. Could you take bread dough and add an insect antifreeze protein to it and allow that bread dough to freeze better in a more consumer-friendly manner? 100%. Is that a GMO bread dough? Not any more than adding flour to that bread dough makes it modified. You're just adding an ingredient. The only thing that you have to go through is you have to show that whatever this ingredient is food safe. And so there's testing to say, okay, it's a cricket protein. Tetrodotoxin is a fish protein. Is it safe to add to produce? No, because it's freaking tetrodotoxin. It's going to shut down all of your calcium channels or sodium channels. So just because it's animal derived doesn't mean it's safe. Snake venom is animal derived, but as long as that cricket protein doesn't end up being a snake venom, then you can add it to whatever you want to add it and make a happier ice cream or a happier bread dough.
Nice. And we had another question from our new friend here. Could those organisms be used as an external thermoregulation patch in some way for maybe long-term food preservation?
So no, they could. Okay, there's no such thing as no. Probably not. But there's an interesting thing that different ice-binding proteins act differently. One of the things that they found in microbial antifreeze proteins is the microbial proteins are actually secreted by the microbe, and they change the ice environment around the microbe. So not just change. So they're freeze-tolerant frogs, and I can't remember what the species is off the top of my head. But you can take. I think it's a wood frog. You can take them, freeze them. You can, and literally there are videos. If you Google freeze a wood frog, and you can see the ice crystals form in this frog. And so those are proteins that are acting within the organism. Some of these microbial proteins are actually acting outside of the organism. So there you very much could imagine some system where we're co-opting these proteins to change the environment. I mean, as a sci-fi geek, one of the things that I'm super interested in is, okay, if we're going to put people in deep sleep or whatever the version is, depending on what the film is, that could involve freezing somebody. You're going to want to freeze them in such a way that you're not shredding the tissues so that when you thaw them out, they're not just a bag of goo. And that would be antifreeze proteins. Oh, we got it?
We got it.
I can thank my ex-wife for this. She worked in a lab that did this work.
They're just kind of spinning the frog around as it's freezing.
Thanks. I hadn't put that together. I do have a PhD in biology.
Not in frog freezing.
No, that was a side hobby. He's going to blink and we're all going to go, ah! This is a really interesting example of the different ways that freezing is handled. So in some organism, the organism freezes and then comes back. In other organisms, the antifreeze proteins keep the organism from freezing. As we've looked more and more at these, the cold-tolerant organisms, we found a greater diversity. And so we found antifreeze proteins in microbes, plants, insects, vertebrates. And the approaches to solving this question of do you freeze, do you not freeze? If you freeze, how do you freeze in such a way you don't get shredded? Seems to differ across different organisms. And there seems to be a huge diversity of approaches in microbes, which is not surprising because there are literally millions of species of microbes. So there are these things called DUFs. And DUFs are domains of unknown function. So one of the really cool things about genetics that will keep me employed until I die is that we don't know what most things do. So we've sequenced the human genome and we've resequenced the human genome. We have certainly hundreds of thousands of versions of the human genome, if not more than that. We still don't know the function of about a third of the genes of the human genome, right? And that's actually fairly profound. Like, we have literally hundreds of thousands of copies of this gene. We know it's a gene. We can predict what the protein is. We can actually put together the structure of the protein, and we have no idea what it does. None. And it ends up in most organisms, this is the case. And when it comes down to it, it's something like 20% of protein domains. So in a protein, you've got this polymer, a string of amino acids. The sequence of that polymer is its primary structure. That primary structure folds up basically either into a helix or a zigzag sheet. That's the secondary structure. And then those secondary structures fold up again to form tertiary structure. Yeah, so we've got examples there. So we can actually predict really well if a primary sequence, that sequence of amino acids, is going to form an alpha helix or a beta sheet or an unstructured domain. Those regions of secondary structure then come together to form tertiary structure. And that's going to form a domain, like a region within a protein. And something like 20% of the domains that are out there don't have a known function. We see them over and over again. So there's something about that structure that seems to have a function, but we don't know what the function is. So there's this domain of unknown function, DUF, creatively named DUF 3494S. And well, the really thing that I think is amazing is people were looking at these antifreeze proteins, and they realized that a lot of these antifreeze proteins have this unknown domain. So it's this overlap between this hole in science where we just don't know things. And it's not just that we don't know what this gene does. We don't know what this protein does. Here's a domain that's found in hundreds, maybe thousands of proteins. We don't even know what the domain does. And this
normally, what is a domain?
So a domain would be like a region of alpha helix and a beta sheet that have come together. And this one in particular is a particular kind of alpha helix and then a beta sheet that's next to it. So those two pieces that are coming together are forming a domain. So a domain is like, if you think of the entire protein, that entire protein's got 300 plus-ish amino acids forming a complex tertiary structure, but you're going to have different domains within that structure. You'll have an active site. You'll have a site over here that maybe is opening and closing over the active site. And a lot of those domains we can put function to, but something like 20% of the domains, we're like, yeah, we don't know. But it keeps showing up. And this one, we've got this DUF, this domain of unknown function, that kept showing up in ice nucleating proteins, these antifreeze proteins. And finally, they're like, apparently, it's an antifreeze domain. So it's just this interesting sort of connection between all these things we don't know. Well, how do you find out the function of unknown things? You study a bunch of unknown things. And so antifreeze proteins are these weird proteins that are involved with ice nucleation. And some ridiculously high fraction of them have this DUF 3949S. And the other way around, I think the sentence in the paper is, at this point, it's not clear that there is a DUF 3994S that is not an antifreeze protein. So the unknowns are what keep us going in science. We just don't know what this gene does. We don't know what this gene does. We don't know how antifreeze proteins work. So there's a lot of work trying to figure out what is it about an antifreeze protein that allows things to either not freeze or to freeze and thaw successfully. Independent from that, we don't know what a lot of protein domains do. And what we have is this congruence between we don't know how these proteins work with we don't know what this domain does. And now we still don't, I don't think, have an exact mechanism, but there's something about this particular combination of an alpha helix and a really funky beta sheet that allows these ice nucleating proteins to work.
A question from chat. Do we know if that unknown region occurs in usually the same similar location? Like, is there any kind of info that we can get in terms of like, well, at least every time this unknown domain shows up, we see this kind of structure?
Right. That's a great point. I don't know that. And you might expect that it would find, so the things that are like associated with being an active site, they tend to happen in the interior, but not too far into the interior of the protein and things like that. So I don't know if there's a region. There was something in that paper that basically I think the idea is this duff must be exposed, because it's likely that what's happening is, if, let's say this is the DUF 3949, the water is interacting with this surface and is nucleating to form a small ice crystal, which is then not able to form a larger ice crystal. So there's something about a surface orientation for that duff that allows that to function. Again, not my field of expertise, but it seemed like a really reasonable explanation.
Cool.
This is great. I mean, I think that one of the punchlines is there's a lot more ways to think about what cryobiology means. And there's certainly some interesting applications. And I don't really think of, I don't necessarily think of applications in science when I think of the utility of science. But it's a non-trivial aspect. And again, if we talk about the idea of outreach, if I wanted to talk to a group that wasn't particularly science oriented, I might say, okay, how many of you would appreciate a better scoop of ice cream? Okay. All right, well, what is a better scoop of ice cream? And I don't think you would get very far in the conversation before the idea of mouthfeel. Like, part of ice cream is really that experience of the ice cream melting in your mouth. We can fundamentally change that with these antifreeze proteins, which we basically are getting because a bunch of nerdy biologists are like, hey, why did that fish not freeze? And from that question, we get a better scoop of ice cream.
It's kind of wild to think about how you can go from a fish not freezing to a better scoop of, like, just that connection, right? And just to illustrate the chat, how many people do you think that goes between? Like, the one that came up with that versus the one that implemented an ice cream? Like, that's not the same person.
No, and I think we're looking at a couple of generations of scientists, because it went from, like, why does this cricket not shred when it freezes, to can we make a better scoop of ice cream? And again, just like an argument for fundamental research, you never know where this research is going to take you. And I do think it's actually worth our time as scientists to be thinking about what are the potential applications. And I think the idea of the ivory tower scientist is not doing ourselves any. So, if I'm really interested in the diversity of ways that organisms respond to their environment, thinking about applications is absolutely something I could be doing.
100%. And thank you, Thomas, for your time today, as always. And thank you, Thomas. Have a good one.